Nematodes are most important component of the soil biomass and play a vital role in soil ecological process. They are involved in many interactions affecting crop plants. Although most of the nematodes are soil dwelling, not all nematode population feed on plant roots directly. As the quote goes, nature does not work with pure culture. Most nematodes either influence or are influenced by associated soil microorganisms. Though plant parasitic nematodes are often considered as plant pathogen in their own rights and are capable of producing disease symptoms. Various associated microorganisms are likely to influence one another as they occupy the same habitat and aggravate the disease. Under natural condition, a plant is a potential host to various microorganisms and they can influence one another as they occupy the same habitat. Since a host is infected by one pathogen, its response to other invaders will be altered. Hence, interaction of plant parasitic nematodes with other plant pathogens, the nature of their associations, their impact on the host and consequential interactive effects on the involved organism may have significant influence upon disease development within a particular host, epidemiology of all hosts involved and ultimately disease management. Infection by one pathogen can alter host response to subsequent infection by another pathogens. Plant parasitic nematodes often play a major role in disease interaction as they are capable of elevating a normally minor pathogens to a major status. Interactions involving nematodes is important because they contribute substantially to variability in plant growth. Plant parasitic nematodes favor the establishment of secondary pathogens, for example, fungi, bacteria, viruses, etc. They alter the host in such a way that the host tissue becomes suitable for colonization by secondary pathogens. The nematodes cause mechanical wounds on host surfaces which favor the entry of other microorganisms. The mechanism of interaction and the precise role of each component in a disease complex are yet to be widely exploited. The fungus nematode, bacterium nematode and the virus nematode interaction are numerous. In some cases, the association of nematode and pathogen breaks the disease resistant in resistant cultivars of crop plants. Weakly parasitic fungal, viral and bacterial parasite can cause considerable damage once they gain entry into plant roots in presence of feeding nematodes. Nematode fungus interaction. Atkinson in the year 1892 was the first to observe the involvement of nematode in disease complexes when he recorded increased severity of cotton wilt caused by Fusarium axisporum, former species was infectum in the presence of root knot nematode. Much of the experimental evidences indicate a biological interaction between nematode and certain soil-borne fungi. In such interaction, nematodes are usually not essential for the establishment and development of fungal pathogens. Instead, the nematode generally and enhance the pathogenicity mechanism of the fungus towards modifying the host plant. For many years, it was thought that nematode wounds facilitate the entry of fungi until a group of scientists led by Ferry demonstrated that cotton roots infested by Melidogene incognita were not attractive for Fusarium axisporum. Since then, the nematode fungus interaction have received considerable attention on important crop like banana, cotton, brinjal, tobacco, tomato and cowpea. Intensity of the damage caused by the nematode fungus disease complex is more severe in plants than their separate infection. Generally, root knot nematode fungus interaction is considered to be one of the important factors responsible for the crop reduction, and very little work has been reported on the nematode fungus interaction and the intensity of damage caused due to nematode fungus disease complex. Nematode fungus interaction of various species can be observed in many host plants. In a similar interaction of Rhizotonia solani and Pratylingus minios and winter wheat, there is a close association between the number of nematodes and the amount of fungus. Full expression of the disease requires effects of both pathogens. Also, an increase in the wheat growth result from a decrease in either pathogens. Interaction of plant parasitic fungi and Pratylingus species have been reported on several plant species including wheat. Pratylingus infection of any root usually result in secondary invasion by other organism. The effect of this secondary pathogen on the plant host vary considerably with the species of host, the pathogen involved and their effect on each other. Of several nematodes of economic importance, root knot nematodes have been most extensively investigated in their interaction with wilt causing or root rotting fungi. In tobacco root is infected with root knot nematode, it makes the roots susceptible for infection from species of Curvularia, Botrytis, Aspergillus and Penicillium. Without nematode infestation, these fungi appear incapable of establishing a parasitic relationship in tobacco. Fusarium solani is associated in the deep root lesion formed by Rhodophilus similis. On the other hand, Rhizotonia solani is found in shallow lesion caused by Helicotylenchus species on banana roots. Semi-penetrant synchriasis root decay by Fusarium species in lemon. Nematode attack on various plant pots produces giant cells resulting from nematode activity which form a suitable substrate for fungal growth, and mycelium readily invade adjacent areas as well. Mechanism of interaction. Nematodes play a supportive role in interaction with fungi, and the nature of support depends upon various factors like nematode species, fungus species and the host plant involved in the interaction. Phytonematodes may play many roles in their interaction with fungi. Wound causing agents. During the process of feeding, all plant parasitic nematodes produce a wound of some sort in the plant host either by simple micropuncture or by rupturing or separating cell. Example, some migratory endoparasitic nematodes like root lesion nematode Pratylingus species cause necrotic lesion or wounds on the host surface that serve as a food base for the establishment of facultative fungal pathogens. Later, the fungus inverts the roots from the site. Lesion caused by Pratylingus penetrans on brinjal and tomato make them susceptible to wilt caused by Verticillium dahlia. Melidogene larvae usually penetrates near the root cap, with subsequent penetration into the meristematic zone of the root apex. Evidence of entry can be observed by dot-like penetration marks. Once the nematode gains entry into the host plant, the larvae move intracellularly without inducing necrosis in host cells surrounding the nematode body. Host modifiers. Several changes are brought about by the plant parasitic nematode in their host tissue as part of the feeding process and derive nourishment from the modified host. Root knot nematodes induce Syngacia primarily in the stelae, which are highly specialized structure. The nematode induced cellular modification causes accumulation of certain chemicals in Syngacia. Another common feature is increase in quantity of organic and inorganic substances like calcium, magnesium, phosphorus, etc. in the galled tissue of affected crops. Differences have been observed in the constitution of giant cell walls compared to those of healthy tissues. Giant cell wall is composed of cellulose and pectin but not lignin. Superin or starch because of the absence of arabinose. This concludes a significant change in pentose metabolism induced by nematode infection. Biochemical changes induced by nematodes are usually favorable to the fungi as it may result in nutritionally improved substrate or there is restriction of certain chemical antagonistic to the pathogen or there might be obstruction in defense reaction by which the host would normally fend off the invaders. Rhizosphere modifiers. Soil flora and fauna are influenced by a wide range of organic compound that exudates from their intact plant roots. The exudates may be a source of nutrients for the microflora or may serve as attractant for the motile stage of the plant pathogen or act as stimulus for the germination of dormant spores. Rupturing of the root cells membrane during feeding, penetration and migration within the root by nematodes directly influence the release of exudates in a quantitative way. There is progressive increase in the levels of electrolytes leaking from nematode infected roots following infection. Mineral and amino acids are the major electrolytes detected on root gall exudates. The concept of nematode induced changes in root exudates relates to the effect on the pathogen in the rhizosphere. The changes in the root exudates resulting in the modification of the rhizosphere has a general influence on the rhizosphere microflora. With this, there is a possibility of a chain of reaction in the nematode fungus disease complex. Resistant breakers. In many instances, root knot nematode turn the otherwise resistant cultivars of plants into susceptible ones. Probably the physiological changes brought about by nematodes as primary pathogens in the host plant are responsible for breaking down the resistance. The nematodes may cause some host response that lowers the natural resistance of the plant to fungal infection. Melidogene incognita and Melidogene capla are known to reduce the resistance to Fusarium wilt in many cultivars of tomato. This breakdown in resistance is considered as a shift in the host parasitic equilibrium due to improved nutrition for fungus, which is making a poor host a good host. Pathogenicity inducing agents. Rarely, nematodes are able to induce pathogenicity in saprophytic soil fungi. Soil inhabiting fungi like Botrtis cinera, Penicillium martensi, and Trichoderma hertianum normally non-pathogenic on tobacco. Invaded tobacco roots infected with Melidogene incognita and caused root damage. Nematode bacterium interaction. Most of the literature available is related to interaction between nematode and fungi. Nematode and bacterium interaction are comparatively fewer than the nematode fungal interaction. Some of the most striking examples in this group include those bacteria which induce wilting in the host, which is one of the primary symptoms. Nematodes may either inhibit the development of bacterial disease or may predispose the plant to bacterial disease. And altogether different disease condition may be produced from the combined effect of both nematode and bacteria than that is caused by either pathogen alone. Sometimes together both nematodes and bacteria result in the production of toxin which might be harmful to mammals. One of the best diseases which have been commonly reported to be associated with the nematode is the bacterial wilt of several host plants caused by Ralstonia solanacearum. Incidence of wilt is higher in presence of nematodes, both Melidogeny hapla and Helicotylengus manus contribute an increase in wilt development. Melidogenic incognita influences bacterial wilt development in tobacco. Another striking example of nematode bacterium complex is the foliage and meristem disorder in strawberry known as cauliflower involving Aphelangides rhizoма bosi and Corynebacterium faeciens. It requires both the pathogen for the expression of complete disease symptom. Different type of interaction between nematodes and bacteria have been recorded. Now, the first one, expression of a disease entirely different from that caused by either pathogen alone. The Dundu or yellow slime rot disease of wheat is a result of essential interaction between Ancuna tritici with the bacterium Clavibacter tritici or otherwise called Corynebacterium tritici. Nematode alone causes ear cockle of wheat, symptoms of which are entirely different from Dundu, similarly cauliflower head. Diseases of strawberry is caused due to interaction between Aphelangides fragriae, Aphelangides rhizoma bosi and bacterium Clavibacter faeciens. In this case, bacteria when present alone has negligible influence. Infection by nematode alone result into weak stunted plants with abnormally narrow leaves. Plants infected with both the organisms show structural deformities in which plants continually produce axillary buds in the ground, giving the appearance of small cauliflower and hence the name cauliflower head. Nematode disposing the plants to bacterial disease. Nematodes may the entry of pathogenic bacteria and damage the host plant by providing them in crust points at the site of nematode penetration. As carriers, vectors are as resistant breakers. Bacterial wilt of coronation increases many-fold in plants infected with Melidogeny or Helicotylengus species. Ground galls in peach and raspberry is caused by Agrobacterium tumefaciens only in presence of root knot nematode. The incidence of wilt in alfalfa caused by Clavifactor michiganense variety insidiosusum is very low generally, but when roots are injured by Melidogeny hapla, the disease increases many-fold. In some incidents, the nematode juveniles carry bacterial cell on their body surface and establish them at the site of infection. Tytylangus dipsaceae is known to carry Pseudomonas fluorescens in garlic, where it causes gaif or light bacteriosis. Similarly, Globodera pallida acts as a carrier for Pseudomonas solanacearum in potato. In case of Dundu disease of wheat, bacterial cells are present on surface of the galls or inside the galls buried in the soil. Here too, the nematodes acts as a vector carrying the bacterium on their surface. Therefore, it is clear that Dundu disease cannot develop without the involvement of nematode. Resistance in potatoes to Pseudomonas solanacearum was broken down in plants infected with Melidogeny incognita agrita. Nematodes as inhibitors of symbiotic rhizobium bacteria. Phytonematodes adversely affect nodulation in plants and play an antagonistic role in symbiotic rhizobium leguminous plant system. Soybean plants infected with Heteroterraclasians race 1 suffer from reduced nodulation by rhizobium that eventually leads to reduced nitrogen fixation. The size of nodule is also drastically reduced in Melidogeny species are known to parasitize nodules themselves and destroy them. Nematode virus interaction. Virus nematode vector relationship are significant aspect of plant pathology. This has raised curiosity in possible relationship between nematode and viruses. Presently, more than 100 species of root ectoparasite belonging to 5 genera, that is Syphinima, Longitudinus, Paralongitudinus, Trichodorus and Paratrichodorus are known to act as vector of virus in plants. Interestingly, all these genera belong to order Torulalimida, have global distribution and moderate to wide host range. Nepo, that is nematode transmitted polyhedral virus particle. The virus particles, they measure 25 to 30 nanometer in size and are polyhedral in shape. Also, they are transmitted by species of Syphinima, Longitudinus and Paralongitudinus. For this example, grapevine fan leaf virus, tomato ring spot virus, that will be called as TRSV, TOBRA or NITU, that means nematode transmitted tubular particles or TOBRA viruses. These are rod shaped viruses which are transmitted by species of Trichodorus and Paratrichodorus. These viruses are categorized into 3 groups and differ in size and host response. They are tobacco rattle virus or otherwise it will be called as TRV, P. early browning virus, that will be called as PEBV, and pepper ring spot virus also called as PRV. Steps involved in virus transmission. When nematodes feeds on roots of virus infected plants, they tend to ingest viruses and become viriliferous. At the time of feeding, the nematodes poke its long odontocystyle style, angiostyle trichodorids into the root. This causes pulsation of esophageal pulp which sucks in the cell sap. In this way, viruses enter nematode body along with cell sap. It should be noted that all 4 juvenile stages of nematodes as well as both male and female nematodes are capable of acquiring and transmitting viruses. Retention. When nematodes do not have access to the host plant, nematode vectors can retain the ingested viruses for long periods. Viruses passes through the stomatium along with the ingested food. They are selectively absorbed on the cuticular lining of stomatium. The so absorbed viruses can retain at different sites depending upon the genera. The ingested viral particle can be retained inside the nematode body for longer periods of time. However, the viral particles are set off along cuticle during molting. Though the viral particles are retained in the nematode body for weeks together, they do not multiply inside the nematode. Dissociation. The viral particles are dissociated from the nematode body when the secretion of esophageal glands pass through stomatium during salivation. Actually, the viral particle slowly gets alienated from the site of retention. All the viral particles are not released simultaneously. Inoculation. When viriliferous nematode penetrates and starts feeding upon the fresh healthy plant, the viruses enter inside the new plant host along with the secretion of esophageal glands. Specificity of transmission. The nematode viral transmission is specific in the sense that all the nematodes cannot transmit all the viruses and vice versa. Example, Syphinema index transmit grapevine fan leaf virus but cannot transmit cherry leaf roll viruses. The chemical nature of lining at the site of retention and chemical nature of protein coat of virus plays a vital role in determining the specificity of viral transmission. Here it is important to note that Tailangites, which constitute major group of plant parasitic nematodes, do not transmit viruses. It could be due to various reasons like very little space available for virus adsorption and dissociation, as the lumen of dorsal esophageal gland empties just posterior to the stylet knobs in Tailangites. Difference in the chemical nature of cuticular lining of Stomatium and contents of Tailangites saliva might inactivate viruses. Now we will see the potential virus vector nematode species prevalent in India. The first vector, Syphinema americanum, transmit cherry rasp leaf, tobacco ring spot, tomato ring spot. The vector Syphinema brevicoli transmit tomato ring spot, and Syphinema davesicadatum transmit Arabis mosaic. Then Syphinema index transmit grapevine fan leaf. Longitudinus elongatus transmit raspberry ring spot, tomato black ring. Paratrichodorus minor transmit pepper ring spot, tobacco rattle virus. Let us summarize the session. Arable soil is a biotic complex in which plant parasitic nematodes share habitat with numerous other microorganisms including fungi, bacteria or viruses. All these microorganisms sharing a common ecosystem in the soil are bound to have some sort of interaction with one or the other cohabitating organism. This interaction may or may not affect each other, but definitely affect the host plant. Among these interaction studies, most well understood are the sedentary root parasitic nematode fungus interaction, foliar nematode bacteria interaction, and virus transmission by terlimate group of nematodes.